In this series of conversations, we'll be discussing global food sustainability with guests who bring a deep understanding of the environmental and cultural challenges facing our society and creative ideas on how to address them. I'm Ash Sweeting and welcome to the Ash Cloud. Today we are joined by Professor Sharon Huws from Queen's University Belfast. Sharon is investigating the intense competition within the rumen microbiome and then using this understanding to create a single solution to two of our greatest food sustainability challenges: reducing livestock methane emissions and maintaining global livestock production.
It's hydrogen warfare that we're talking about, and actually you can do something with hydrogen. So we just need to redirect it into the correct pathways. I say we just need to. Of course, it's not something that happens overnight.
It's all about the hydrogen.
So if you can shift the biochemistry a little so that some of that hydrogen is shuttled to energy, that will also reduce methanogenesis because there'll be less hydrogen available.
Sharon is optimistic that a truly sustainable solution exists that both improves farm productivity and decreases methane emissions.
If we can reduce methane and also redirect the hydrogen into an energy source, that's hugely beneficial. So that's what we're trying to do, find ways in which we can capture that unused hydrogen as well, so that you get more meat or you get more milk.
Solutions that only focus on methane and ignore hydrogen have limitations.
Because if you have too much hydrogen buildup in the rumen in particular, you will see some issues coming in. So you'll see a change in that rumen microbiome, and there might be some health issues that come in for the animal.
Only focusing on methane also has economic and farmer adoption challenges.
You know, ultimately, reducing methane, if you haven't got a production value, is all about who pays, because someone has to pay ultimately.
Next generation sequencing technologies are game changing.
We use Omics technologies quite often, so there'll be sequencing technologies so that we can understand what makes an animal a low methane emitter in the first place. You know, what microbes do they have? Because it's highly likely that they have an abundance of bacteria in particular that can act as a hydrogen sink. So they shuttle the hydrogen away from methanogenesis.
Early results are looking positive.
But certainly some of the lactic acid bacteria are showing anything up to about 20% reductions in methane emissions.
Nature continues to provide a wealth of knowledge and guidance.
And I think we have a lot to learn from termites, from wallabies and from kangaroos in that nature, because they produce very little methane. And the reason that they produce little methane, just a little bit of methane, is because they have reductive acetogens that take the hydrogen away.
Sharon, thank you very, very much for joining me today.
No problem at all. Very pleased to be here.
So we're here today to, I guess, investigate the issues around livestock methane emissions and what can be done about them. And from my perspective, the methane isn't actually an issue for the animal. The methane is the animal's attempt or the animal's process to get rid of the excess hydrogen within the rumen. So do you want to dig into that side of things a bit more for us, please?
Sure, no problem. Yes, it's a process. Methanogenesis is a process that's evolved over many, many years, and it's very natural for those ruminants to want to get rid of hydrogen, because if you have too much hydrogen buildup in the rumen in particular, you will see some issues coming in. So you'll see a change in that rumen microbiome, and there might be some health issues that come in for the animal. So it's a good thing to remove the hydrogen that's in the rumen fluid.
And do you want to, what other options are there to removing the hydrogen other than methane?
Yeah. So when an animal eats the forage, that forage is full of carbohydrates. So they break it, as they break it down, they produce hydrogen, which is used by the methanogens. But also on the other side, they also produce the volatile fatty acids, and they give the animal their energy. So, and in the production of some of those, we call them VFAs for short, so I'll call them VFAs. They also utilize some hydrogen to make some of them, like propionate, for example. So if you can shift the biochemistry a little so that some of that hydrogen is shuttled to energy, that will also reduce methanogenesis because there'll be less hydrogen available.
Excellent. Well, that sounds like a potential win-win because you're capturing the energy inside the animal as well as mitigating the climate side of things. Do you want to tell us what you're doing in that space?
Yes, of course, no problem. So we work with a range of dietary interventions to reduce methane emissions. So, and most of them actually reduce the process of methanogenesis. But in actual fact, when you reduce that pathway, you end up with quite a lot of hydrogen. And in fact, at the minute, the farmer, in most places, they get paid for production and not for reducing methane. So in fact, if we can reduce methane and also redirect the hydrogen into an energy source, that's hugely beneficial. So that's what we're trying to do, find ways in which we can capture that unused hydrogen as well, so that you get more meat or you get more milk.
And what tools are you? Well, do you want to just outline the process?
Sure.
One of the things, and this is, correct me if my understanding of this is not 100% correct, but if you look at cancer treatments over the last 10 or 15 years and how they've been revolutionized through the next generation genomic sequencing technology, and people that had a diagnosis that even 4 or 5 years ago would have meant they only had weeks or months to live, are now having longer and much more productive lives and much worse outcomes. And from my understanding, what you're looking at doing is pointing some of that same technology at the rumen microbiome to actually understand what's going on. Is that correct?
Yeah, that's true. So what we use omic technologies quite often. So there'll be sequencing technologies so that we can understand what makes an animal a low methane emitter in the first place. What microbes do they have? Because it's highly likely that they have an abundance of bacteria in particular that can act as a hydrogen sink. So they shuttle the hydrogen away from methanogenesis. So that those technologies have helped us a lot. But we're also increasingly going into what's called culturomic technologies. So we actually isolate pure cultures of some of those rumen microbes, particularly bacteria, and look at their capacity to act as hydrogen sinks. So how well do they use hydrogen in the process of making a VFA? And then those candidates, they can be used almost like a probiotic. So a probiotic alongside a mitigation measure that might also reduce methanogenesis will give you enhanced methane reductions, but the animal will also produce more.
So I guess solving the problem because if we want to solve the climate problem, we need to look beyond just the methane. We need to look at what the issue for the animal is, and if by solving the animal's problem, then we'll solve the climate problem at the same time. Is that
Absolutely. I always use this strap line, that rumen microbiome is really important for planetary health. And that is for the health of the animal, for the health of the products that are produced for human consumption, and for environmental health. But if we look at just the axis of animal health and environmental health, if we have a healthy animal that has a low infection burden, for example, a low parasitic load, they're going to produce more of the meat and the milk, and ultimately, in their lifetime, they're consequently producing less methane. So that whole picture, you can't look at one thing separate to all of the others.
And whereabouts are you in this whole process in terms of, having something that would be ready to be used on farms, either in a research capacity or more broadly?
Yeah, so there is one company in particular that has a product that's now approved in many countries, so they're probably at the forefront, and I think it's perfectly fine for me to say that it's Bovia, and they'd probably welcome me saying that, which results in about 30% reductions in methane emissions. But one of the challenges that they have now is that they don't get enhanced production. So you have to look at things like direct fed microbials or probiotics, whatever you want to call them, in order to act as hydrogen sinks. And I guess, that's all very dependent on whether countries put a carbon framework in place. Ultimately, reducing methane, if you haven't got a production value, is all about who pays. Because someone has to pay ultimately, because the farmers obviously can't pay for that product. So it has to be consumer or a government-led carbon framework. But there are others as well. There's things that we work on, like the red macroalgae, Asparagopsis taxiformis, which does seem to have a little bit of a production benefit as well. So that data is quite fresh. It's from herd level data, that it's not our own data at the minute. And there are other products, like Asparagopsis was probably the next one to Bovia in terms of being ready for the market. And then there's a whole number of other products which are, I would say, are lower down in their readiness. So in their, I call it the technology readiness level, for example.
And what about the work that you're doing and the Omics work that you're doing and the direct feed microbials or probiotics that you're looking at? How far away, where are you at in that whole stage of research?
Yeah, I think this is a really exciting space. We have a few novel isolates. So our whole culturomic approach has isolated quite a lot of novel bacteria that may be even better than what we have already, potentially, in order to redirect hydrogen. But we're right at the very beginning of that. We're doing the in vitros in the lab to try and simulate what might happen in the animal. But I think that area is really promising. And very recently, we have a new flagship project. So this is a Global Research Alliance flagship project. So for those of you who may not know about the GRA, they're actually a massive consortia of 66 countries, and lots of bodies like the FAO are involved as well. So they gave us a flagship status for our culturomic program, which has meant that we have been able maybe to get a little bit more funding around isolating more novel rumen bacteria, archaea, and fungi as well, because there's a whole wealth of genetic diversity in there that we're really not tapping into. Particularly if you take a sample from a low methane emitting animal, can you get the particular isolates out that are responsible for redirecting that hydrogen? Because they should be one of the best direct fed microbials. So we're at the beginning, but we're also got this huge, huge global project where people across the world will be culturing novel isolates. So we're really hopeful that we'll be able to tap into some of that novelty.
Okay, well, congratulations firstly on being recognised that way and having that acknowledgement, I guess, and hopefully some funding to go with that. What, I guess, if I had the blank check to write you and said, okay, what would you do? What would you do?
What would I do? So in that global project, that's exactly what I would do, although I could do with more funding on top of it. So what we have is we have about 13 culture hubs across the world. So they're universities or institutes that have the infrastructure and the capacity to culture from samples that they have or samples which are sent to them from other countries. In particular, we want samples from the global south because they will be fed, the animals will be fed an array of different tropical feeds. So the microbiome will be slightly different. So all of these hubs, including ourselves, who are the lead hub in Queen's University Belfast, will all be culturing those microbes. Most of us are concentrating on bacteria, but other places have capacity to do for the fungi and the archaea as well. So our aim is that we should have about 2,000 to 3,000 minimum novel isolates by the end of the 3-year project. And even that is the tip of the iceberg, because if we had another 3 years, we would probably be able to close quite a lot of the gap in what we have in culture compared to what lives in the rumen. And our intention is those microbes are going to be available in culture collections. So if anybody has anything that might have potential IP, we're asking people not to send them, because there's nothing more frustrating than to have a genome and not be able to get to the isolate in order to test them. But in that project, we're also going to be looking at what VFAs they have the capacity to produce. And in particular, we want to look at the ability to make propionate or to reduce acetate, because they're the main areas where you can really redirect your hydrogen.
So there's a couple of things there. Firstly, I guess, is how different is I guess from perspective, you said you're going to be having hopefully 2,000 after 3 years. And just for people who might not know as much about the rumen microbiome, is that 20%, 5%, 1%, 0.1% of what's in there? And then how much does it vary between, you're in Ireland, I'm in California, but Australia or Africa or South Asia, or South America? How much does it vary?
So at the minute, just to put it into perspective, we have somewhere around 500 microbial genomes for the rumen. So we're talking about adding 2,000 to 3,000 on top of that. And we think by doing that, we should have somewhere in the order of 60 to 70% of the rumen microbiome covered. And that we make those calculations because there was a global study that used ribosomal sequencing to see what is normal for the rumen across the world. So there were samples taken from different ruminants across the world. So from our pure cultures, what we can do is map back to see the matches, and then you get an idea of your coverage, if you see what I mean. But we think we can easily get to about 70% in 3 years' time. And to answer that question on variation, I think I have inadvertently answered that, because that rumen census piece of work that looked at what's normal showed that there is a core microbiome, whether you're talking about, a goat or a cow, for example. There is a core that you will always find in those ruminants. But then the breed, and in particular, the diet, defines what the rest looks like. And that study showed that it was really mainly diet that drove that differential from the core.
Okay, and because you're gathering the genomic information from these microbes, so are you looking at trying to identify the actual genes that are the hydrogen utilizers? And so when you have a genome, you can say that one we believe will be a hydrogen utilizer, so you can actually...
Yes. Yeah, yes is the answer to that. So when we have these genomes, we will want to dig into them as much as possible to get the genetic understanding of which genes are responsible. Of course, whether we can do that in a 3-year project is probably no. We'll probably get to the point where we have all these isolates, we'll have them characterized, we'll have them genome sequenced. But whether we have actually, it's very unlikely that we will have mined those genomes fully.
I guess the next question is about the competitive nature, because just because you have something, a bug that's been isolated and cultured that's might be a wonder bug, how do you get it to survive and persist and be competitive within the microbiome? Or is that a space that we also need to spend some effort investigating?
Yeah, well, obviously isolating from the rumen itself is really helpful, because you know that they can survive in there. In particular, if you take some of the most dominant species that naturally prevail in there and can redirect hydrogen, that's useful. But also we have to look at encapsulation technologies, because of course you have to grow them at scale, in large scale. They're strict anaerobes, and there has to be a way to encapsulate them and keep the oxygen out. And at the same time, when they're ingested by the animal, they can then be released slowly as required and not in one go. So there is a technology has to move at the same time, if you like.
And what are the challenges there, or where are we at as far as moving along that side of the whole process?
I think that area is really moving forward. It is moving forward, which is why I think direct-fed microbials have almost had an upsurgence recently, because the technology is there to be tested. And I think in the same way, when we look at our additives as well to reduce methane, the technology is being pursued in order to release it slowly into the rumen of a grazing animal. So for grazing animals, you can't get those additives in very easily because they're always on pasture. So we have to have those mechanisms of releasing slowly. So there's a parallel there with what's required on the direct fed microbial side. But the technology has advanced substantially.
That's wonderful to hear, because I think it's easy to have a picture of livestock methane emissions and think of a production facility in Europe or in North America or in Australia. However, when you actually look at the numbers, the two countries with the largest cattle populations are China and India, and then you've also got hundreds of millions of animals in South America and also in Africa that are much more dispersed and harder to get to from a product perspective. So having something that would work in those would be a big game changer in terms of global reduction of emissions.
That's very true, but I think we have to be pragmatic when we talk about some of the countries in the global south. You know, the infrastructure and the cost of some of these technologies at the minute would be very inhibitory. But of course, when you look at South Africa, for example, there are lots of things that they can do in their management, for example, to make those animals more productive. But we have to start somewhere. We have to start, you know, in countries that have the infrastructure, and then help all of the other countries move towards these technologies as much as we can.
So you mentioned earlier that by capturing that hydrogen energy within the rumen, you get a potential production benefit for the farmer. Would you be bold enough to predict what may be possible in that area?
Yes, I think I can predict, because there's more and more animal level data coming out at the minute. So bear in mind that this is with isolates that we already have, but certainly some of the lactic acid bacteria are showing anything up to about 20% reductions in methane emissions. And that data is them alone. It's not them in conjunction with an additive like Bovia, for example, where you should have, well, as the name suggests, an additive effect of both of them.
Mm-hmm. So if you get rid of the lactic acid, then that's also going to help with reducing methane. Is that correct? Is that what I'm understanding, or is that?
Well, lactate is a precursor for propionate. So, and in some of those studies, they've actually made the silage using a lactic acid bacteria to help preserve the grass. And then when it goes into the rumen, they're seeing benefits in terms of methane, because they're producing lactate, which is converted into propionate. And that conversion actually utilizes hydrogen.
Okay, so the method of interaction or not of implementation could be within a silage or something like that, as well as actually giving it directly to the animal.
Yes. Yeah, that's absolutely true. And, you know, lactic acid bacterium is just an example, and they're facultative anaerobes. So they're also slightly easier to deal with when they're not strict anaerobes, as you know.
Yeah, no, that makes a lot of sense.
So yes, people are looking at more practical ways as well. What I would say in all of that is that it's likely to be the tip of the iceberg, because the diversity that we have can probably do even better if we manage to get it out into culture.
Going forward, maybe probably 10, 15 years' time, do you think it would be likely that someone would have a herd of animals and they'd have their rumen microbiome tested, and then essentially there'd be a prescription of various different bugs or microbes that they would be given? So you're actually tweaking the microbiome at that level. Is that the sort of ultimate aim of where you're going?
That's quite possible, because also by having all of these cultures, what we're trying to do is enable the ability to just look at diversity of who's there, and then to know the function, so that the inferred function is very accurate, if you see what I mean. Because if you have the isolates, you can, you know what they're doing in essence, and then you can just look at diversity in the rumen, which will be enough. And that, of course, is much easier to do. You can look at diversity very simply. So people would be able to take some samples potentially and know, okay, what is wrong? Is this good or is it not? I mean, the only thing that I would say from that is, of course, you can't get rumen samples easily. It is an invasive procedure, and you might want to perhaps, the fecal samples are not a good proxy in themselves, but you'd have to think about
whether you could find some other proxy or some other way of analyzing that. There's
Yes.
been people I've spoken to who there are across nature, there are herbivores that are non-methanogenic. There's a lot of variation there. Where
Yeah.
do you see, I guess, cross species um, coming into it from both
Yes.
a research perspective in terms of better understanding what's going on, and then
Yeah.
a intervention perspective?
I'm smiling because that's a very, very good question. And I think we have a lot to learn from termites, from wallabies, and from kangaroos in that nature, because they produce very little methane. And the reason that they produce little methane, just a little bit of methane, is because they have reductive acetogens that take the hydrogen away. So acetogenesis and methanogenesis compete in any environment. So in the rumen, because methanogenesis is higher, then your acetogenesis is always lower. So your ability to move your hydrogen into reductive acetogenesis is dependent on whether you can lower the methanogenesis.
Yeah.
But in animals like termites or, well, vertebrates and wallabies and kangaroos, the converse is true. So reductive acetogenesis outcompetes methanogenesis nearly always. And some of those microbes could be particularly key for use in the rumen. Just because they come from other animals doesn't mean to say that they wouldn't be suited for the rumen, because many of those acetogens are ones that you would see in the rumen, but they might have a little bit more competitive advantage that they can suppress methanogenesis.
Have you ever thought, or do you understand, or I'm thinking about this from an evolutionary perspective, why we've got this same issue of hydrogen, and there's been different evolutionary paths, and obviously there must be some competitive advantage in the different paths because that's what's evolved. Are you aware of, I guess, this from an evolutionary perspective and how that sits?
Yeah, absolutely. So utilizing hydrogen is a way of gaining energy. So it's you gain ATP by making all of those, which is an energy source. And I think that this is what fascinates me the most about that rumen. It's the chemical warfare that actually goes on because they're competing to make that energy. So essentially they're utilizing the hydrogen as much as possible, but you will also get some microbes that produce antimicrobials. So they produce antimicrobials so that they can really compete in the rumen. And that's the hypothesis that some scientists have used when they've used lactic acid bacteria to redirect hydrogen. They have chosen the lactic acid bacteria that produce the most antimicrobials, because the thinking is that when they hit the rumen, they will have more competitive advantage because they have this cascade of antimicrobials, allowing them to compete. But it's that ecology of how they're all working together and how they compete that it really fascinates me in that whole war. It's a warfare around hydrogen.
That is absolutely fascinating. Do you think because everyone, as human society, we see the problem as methane, and so there's all this talk in the media and in governments about methane, and the issue is actually the hydrogen. And do you think the fact that the conversations around the wrong topic means that resources, energy aren't directed as beneficially as they could be?
Yeah, I think so. I think the whole space is seen very negatively. We've all seen that in the media. We see various countries like Northern Ireland here have put a separate 46% reduction needed in methane by 2050. But I think ultimately, the conversations with the scientists are perhaps not happening as much as they should, to get this real understanding that it's hydrogen warfare that we're talking about. And actually, you can do something with hydrogen. So we just need to redirect it into the correct pathways. I say we just need to. Of course, it's not something that happens overnight. But I do think, these conversations, and also how we talk about it in a way that it makes sense, that we're not, as scientists, really overcomplicating things, that people are immediately shut off from what we're saying, is really important.
Well, that's 100% the reason why I am speaking to you today, and have spoken to, many of the other people I've spoken to previously on the podcast. So thank you very much.
Not a problem. I do like to talk about this topic very much.
And is there anything else that we haven't discussed that you think should be added?
No, I think it's really important that the outlook is really positive. If you look at the press, for example, you see a lot of negativity, but we're already on the path in reducing methane substantially. So I think it's really important for us to convey that, that it isn't we will have to cull all of the herds, etc. That absolutely the solutions are in development, and some of them are nearly ready.
Excellent. Well, thank you very, very much.
No problem. Thank you.
You've been listening to The Ash Cloud with me, Ash Sweeting, in conversation with Sharon Huws, recorded in California in October 2022.